afternoon. Thank you to the committee for the opportunity for our group. And I'm going to present our in-progress work on viral strain networks tracing human or environment-linked hepatitis C virus dispersion in France and in Europe, we hope. And our group is involved not only in medical virology, but also in basic research in virology, combining our medical staff for the virologists at the hospital and a group in basic science on waste water-based epidemiology. And hepatitis E virus is one model for our emerging viruses coming from the zoonotic world. Is it? Which button is it? And as you can see, we have with hepatitis C virus a lot of questions. The first is the seroprevalence. We are not completely sure that our seroprevalence data is a good evaluation. We believe that we are around 12%. And around 20 million a year of HIV infections, and with more than 40,000 deaths evaluated in 2015. But a lot of questions on this. And concerning the transmission routes, it's the first goal to understand how to prevent everything. HIV infection in Europe could be not only due to interhuman and interspecies from animals contaminations, but maybe could be also water linked, such as it is observed in low-income countries. Concerning our material and methods for this part of the work, which is in progress, we have first collected European sequences from public database to perform classical phylogeny on one side, and on the other side to perform a modelization to highlight HIV transmission networks. This approach by the collaboration with the mathematics has been very interesting on the SARS-CoV-2 model, but we have, as you know, a lot of data for COVID-19. It's quite more difficult for HIV. And this first part is to evaluate the fitness of HIV clones by their clustering according to time and to viral amino acid composition. And for the second part of our work, we are working on HIV subnetworks. This is to highlight functional hotspots with molecular patterns within the proteins, which could facilitate the transmission and the fitness inside infected patients, infected animals, but also which could influence the behavior of the virus in the abiotic environment outside the bodies. So for the first part of our in-progress results, this is a distribution of the viruses according to one part of HIV of 1, the methyltransferase gene. This is a gene which is quite easily complete in the database because it's within the 5' extremity of the genome, and we have regularly successful PCRs in this site. And as you can see here, you have a distribution according to time and amino acid of this methyltransferase gene. And as you can see, I hope you have either human sequences as the circles and he also animal strains as squares, and from 301 sequences from the humans and 79 sequences from animals in this example. And the sequences appear to be randomly distributed. So nothing to help us to understand the distribution of the sequences. With this second graph, more importantly, we decided this time to analyze the sequence distribution according to a gap we had observed in the methyltransferase at the position 135. And you can see it's the same organization of the graph, circles for the humans, squares for the animals, but the colors this time, it's not according to time, it's according to gap. They are observed in human strains, no gap in green, and it's observed not only in humans, but also in animals. No any gap at this position for the animals. And you can see that the blue cluster, the sequences with a gap in human HIV origins are very clearly not randomly distributed, but grouped. And this could maybe be linked to the gap at a proline residue next to the catalyte seed of the enzyme, and this is under exploration. The second part of the results and of the work was to focus on the surface proteins of the virus of 2, the capsid protein, and of 3, which is a specific phosphoprotein interacting with OF2. So I will not develop the details because of the time, but we have chosen a very interesting review meta-analysis, which was available in the public literature, published in 2022 by Takrisu et al., because the keywords, which is very interesting in our groups, is water sling. And these data came from human animals and sometimes from water potentially source. So for this part of the study, I will go first on this slide. You have a classical phylogeny which is performed on the left by our own French sequences obtained in the northeast of France in Nancy group, 48, and with the additional sequences just after, 50 sequences were obtained by combining our sequences to the sequences published in the meta-analysis. And to summarize the combinations of the exploration of the roots of the virus, you can see that this combination of results, I will not develop all the details, but can allow us, for example, in the black part of the phylogeny tree on the right of the slide, we can observe that some rabbit related strains observed not only in the animals but also in our sequences had been observed in Toulouse, France. And for example, in the blue part of the phylogeny tree, we are allowed to suggest that one of our sequence was very next of a sequence from a Spanish pig. So we are usually observing this trees, but we believe that it will be more often done to a better understanding of these transmission routes. Sorry for the button, it's on my friend. And for the last part of the analysis, Yes. So to go to the conclusion, the other analyses are on specific modifications of the amino acid. And with alpha-4-3, we can observe that one modification in amino acid in OF2 in the capsid can modify with a very strong impact the structure by helix and will modify also the interaction of the external part of the OF2 capsid protein. And we can, yes, so I will go to the conclusion if I can. Yes, and these results can help us to understand the transmission networks in Europe by pulling our data quite regularly, by keeping some very interesting molecular signatures either in the enzyme or in the structural proteins to, with this K-functional amino acid, not only help us to understand the transmission.
Thank you very much. We'll close here, but thank you.
Okay, thank you.
Please join us.